Evrony. He is an assistant professor at NYU School of Medicine Center for Human Genetics and Genomics in the Department of Pediatrics and Neuroscience and Physiology. He received his undergrad degree in brain and cocci from MIT and completed an MD PhD at Harvard Medical School. In his graduate research, he led the development of single-cell genomics technology that discovered diverse somatic mutation in human brain, which he's also going to be talking about this today. And for this work, he was recognized for several awards, including the Eppendorf Science Magazine Prize for Neurobiology, the MIT Technology Review Top Innovators Under 35 Award, and the Pew Foundation Biomedical Scholar Award. He completed pediatrics residency at Mount Sinai Hospital and subsequently launched his laboratory at NYU. And in addition to his basic research on somatic mutations, he also founded and directs an undiagnosed disease program that identifies genetic diagnosis for children with severe illness that are unsolved despite extensive prior medical evaluations. Yeah, welcome, Gilad.
Thank you so much to everyone, and I'm really pleased to present to you. As you'll see, there's actually roots to this work historically going back to UCSD, as well as ongoing work that I admire going on at UCSD in this field. So I hope many of you find this interesting, and happy to take questions at any point. This overall theme of research in my lab is motivated by this really remarkable and fundamental observation that all cells in our body, including healthy cells, every healthy cell, accumulates DNA mutations throughout life. And that's schematized here. With age, you get an increase in mutations that actually turns out to be linear the way it's schematized here. And this has been shown over and over in many different ways in every tissue that's been examined to date. There's one study here using an early form of error-corrected sequencing that showed this accumulation in bulk brain tissue, in the kidney and colonic epithelium. Single-cell genomics that I was involved with, and later on others as well, has shown this accumulation with age. Other methods have shown this in epithelial tissues, and esophagus and endometrium shown here. And we actually also have, as a field in genetics, have known this by the observation that the number of de novo mutations in children correlates with the age of the parent at conception, both paternal age and to a lesser degree maternal age, which implies that germ cells even accumulate mutations with age, including eggs. Really, there seems to be no cell that escapes this phenomenon. I should even mention that this phenomenon was observed through even more non-genomics classical methods, even as early as 40 years ago. So it's not just in the era of sequencing that this has been observed, but even before then, using various selectable markers and leukocytes, this was already shown. It's just that in the past 10 years, genomics methods have really shown this to be true across many tissues and have actually taught us more about the patterns of these. A secondary motivation for this particular project I'm going to describe is that when we say that mutations accumulate in healthy tissues, what we mean by mutations, to be clear, is changes that are present in both of the strands of the DNA double helix. But of course, lightning doesn't all of a sudden strike twice in both strands somehow miraculously, but rather all mutations begin, almost all mutations begin first as some kind of damage or change, a mismatch in one of the two strands of the DNA double helix, which if unrepaired or misrepaired, then can, and with some fraction of the time, be transformed, or as we say in genetics, fixed into a mutation that's present in both of the strands. So if we really want to understand the origins of these mosaic somatic mutations that accumulate in healthy tissues, we'd like to also observe these earlier precursor changes. So a little bit of history, especially for the crowd here at UCSD, which is how did I, and I would say a lot of the field, become interested in this idea in the age of genomics. And it was actually a lot of the motivation came from Rusty Gage's lab at the Salk Institute and others at UCSD, which this important observation that neural progenitor cells seem to be capable of undergoing retrotransposition by L1 elements. And then that motivated our group back when I was in the lab of Chris Walsh and others subsequently to confirm this in humans and then expand from there to other types of somatic mosaic mutations. And turns out, of course, not just L1, but pretty much any type of mutation it seems to be occurring during brain development and also later with age. This is one example from my work back at that early stages where I developed single-cell genomics methods. We were able to indeed identify two L1 retrotransposons that occurred during brain development. One of them colored in blue, which was mapped to just one gyrus of the brain, another in green that seemed to have spread at low mosaicism across an entire hemisphere, and it was absent from non-brain tissues. And so we sort of could estimate when these may have occurred during brain development. And of course, these somatic mutations that I've been discussing don't only occur during development, but also after development, even in post-mitotic cells, we now know. And this is sort of a shout out to Joe's group, who has also contributed very significantly to this. Joe's Gleason's group recently showed that using these developmental somatic mosaic mutations, you can actually start to time when developmental splits happen. For example, the split between the left and the right hemispheres seems to be occurring early as the first developmental split before sort of anterior-posterior developmental splits. So you can actually use somatic mosaic mutations, not just inherently they're interesting because they can lead to disease, but as a tool for understanding how the brain and actually how development across all organs is occurring in humans. So these are naturally occurring lineage markers. And so things that you could only ever dream about doing in humans, turns out now you can do them without anything invasive like retroviral markers that you introduce or dyes, things that are obviously unethical to do in humans. So now that the field has sort of examined somatic mutations across many tissues and also in cancer, there are ways now to look at the patterns of them and try to deconvolute them to different processes. In other words, the mutations don't seem to be occurring completely randomly. There's biases in terms of their sequence contexts. And if you could sort of pull together sets of mutations that share certain contexts, sequence contexts across many samples, that implies there's some specific process giving rise to that pattern of mutations. And so far, there are two patterns or mutational processes, other people call them signatures, that seem to be ubiquitous and also seem to be occurring with age. And that is a signature SBS1. SBS stands for single base substitution 1, and signature SBS5. Signature SBS1, we know very, very likely what the cause of it is because it's C to T changes at CG dinucleotides. So it's very likely due to deamination of 5-methylcytosine. In other words, SBS1 is a consequence of DNA methylation. The methylation of our genome, which is important for cell state specification, gene expression control, itself is mutagenic, and there's sort of like an unfortunate byproduct of that. Interestingly, SBS5, which accounts for, in most tissues, about 80% of the mutations, we don't know, nobody knows its etiology. And you can see here in samples from the colonic epithelium that the burden of these of mutations attributed to these two signatures increases with age. So again, we'd like to know the precursors of these mutational processes, be able to observe them, and indeed try to find the precursors of all types of somatic mosaic mutations at the single strand level. I mentioned briefly that somatic mosaic mutations can be used as a tool for understanding development, but of course, they're also a very important cause of disease. Cancer is the obvious one. cancer is essentially a somatic mosaic genetic disease, but there are other non-cancer diseases, and I was involved in one such category of diseases and identifying somatic mosaic mutations in them. And again, Joe Gleason's group, concurrently to my work with Ann Poduri and in Chris Walsh's lab, also identified this, which is that somatic mosaic mutations in the PI3K mTOR pathway cause focal brain malformations. And this was the first case that we had studied, and I applied the single cell genomics methods that I developed to this first case, and indeed we found that the causative somatic mosaic mutation in AKT3 was present both in neuronal and non-neuronal cells, indicating it occurred early in brain development before in neural progenitors that could still give rise to both neurons and non-neuronal cells. Later, in residency, when I became interested in applying some of these methods to unsolved clinical cases, I identified a case with a mosaic mutation in JAK1 that was an activating mutation that led to autoinflammatory disorder that was actually treatable with the drug tofacitinib. And this girl had resolution of all of her autoinflammatory severe symptoms within a few weeks of going on this drug after 18 years of no diagnosis. There are many other kinds of non-cancer somatic mosaic diseases, and I think many more to be discovered, actually. So the open key questions are, what are the single-strand origins of these mosaic mutations? How do somatic mutation processes vary across organs, cell types, and environmental exposures? Looking across all studies to date, it does become apparent that the rates and patterns of these mosaic mutations are not exactly the same across different organs and different cell types, but it's still an understudied question. We'd like to know, as a field, I think, how these patterns vary across the population. Are they predictive of cancer risk, possibly? And there's a long-standing hypothesis going all the way back to Leo Szilard in the more than 50 years ago, which is that the accumulation of DNA damage and mutations may actually be one of the causes of physiologic aging. And this is still an open question. So answering all of these questions requires technologies that can sequence single molecules of DNA accurately and also scalably. So you can do it in many samples. So why has this been a challenge? And actually, it took some time after the commercialization and invention of next generation sequencing technologies to even be able to see mosaic mutations accurately. And that's because standard next-gen sequencing has error rates of about 1 in 10,000 bases. So every about 10,000 bases that are sequenced, there's an error, an artifact. But the level of these somatic mutations I've been talking about are only at about 1 in 10 million to 1 in 100 million bases. So essentially the noise is higher than the signal. It's a classic signal noise discrimination problem. You can't detect a signal that's way below, that's a much lower level than the noise. And so specialized methods are required. So all those studies I showed you before, there was some aspect to them that made them different from standard DNA sequencing. And this is sort of an overview of those methods. So single cell sequencing, which I mentioned, and that's one such approach. And as I mentioned, I have a soft spot for this approach because I developed this method back when I was in the lab of Chris Walsh. And I was personally motivated by decades of hypotheses that somatic mutations may generate functional diversity in the brain, which I mentioned. Rusty Gage's group and Alison Wootry really raised in the field as an exciting research direction. And so at least when I set out to do this 15 years ago in 2009, it wasn't known whether single-cell whole genome profiling would even be feasible, but fortunately it worked. And this approach has since been used to discover diverse types of somatic mutations in the brain and other tissues, as I mentioned. Another class of special genomics methods that have taught us a lot about these somatic mutations is duplex sequencing, developed at the University of Washington out of Larry Loeb's group originally. with additional improvements subsequently by the lab of Inigo Martin Corraine at the Sanger Institute. This method works by attaching special barcodes to double-stranded DNA molecules where the barcodes are reverse complements of each other, so that later, when you sequence many copies of the original top strand and many copies of the original bottom strand, you can use these barcodes to figure out which reads came from the top strand and which from the bottom strand of the original double-stranded DNA molecule. And you can then computationally pair together a consensus sequence of the top strand and the bottom strand and basically filter out any changes that are in only one of the two strands of the DNA. And that has a very powerful filter effect that allows it to identify double-strand mutations extremely accurately with an error rate less than about 1 in a billion. So essentially the noise level is brought down. Another approach is sequencing microdissected clonally related cells. So in epithelial tissues, especially in other tissues where the cells that are clonally related are physically next to each other, remain physically next to each other, you can dissect out a group of clonally related cells, and the somatic mutations that they share, because they're closely clonally related, can now be detected. Essentially, their signal is higher than it would normally be. However, an important sort of limitation of all of these methods so far is that they do not interrogate the original DNA molecules directly. And this has actually precluded the fidelity necessary to see these single strand changes that precede double strand mutations. All these methods essentially amplify the DNA either on or off the sequencer. So my lab set out to see, can we reach these super high accuracy error levels that we need without any amplification of the DNA before it's sequenced. So essentially interrogating the original DNA molecule directly. So we knew we needed some kind of single molecule sequencing technology, which is basically either PacBio sequencing or Oxford Nanopore sequencing. But both of those commercial instruments, their marketed error rates are nowhere near what we would need in order to detect not only double strand mutations, but single strand changes. So we thought, is there a way to modify how we use these commercial single molecule sequencers to increase their fidelity, not just tenfold or 1,000-fold. Really, we had to increase their fidelity by 1,000,000-fold at least in order to bring the noise level down below the true signal of somatic mutations. We thought that PacBio sequencing might be able to do this because it actually reads an individual molecule several times, normally about 5 times. The way PacBio sequencing works is a double-stranded DNA molecule is circularized with a hairpin adapter, as you see here, and the polymerase that reads this DNA molecule goes around and around the molecule over and over, reading the top strand and the bottom strand and the top strand and the bottom strand many times. Well, actually, ordinarily only about 5 times. And then these random errors in red can be filtered out. So we thought, what if we could increase the number of times we go around the molecule by using smaller DNA fragments than usual, not 20 kilobases, but 1 to 4 kilobases. That would allow us to get closer to 20 passes around the molecule. And really, the fundamental principle is repeated measurement. Just like in any scientific experiment, the more times you repeat an experiment, the more confident your estimate is of what you're trying to measure. Same exact principle here. And in fact, similar to any scientific experiment, generally error, if the error pattern is random, you actually get exponentially increasing fidelity. And so it turns out we thought it would only be necessary to have smaller DNA fragments initially, but then we realized that actually by analyzing the data, it exposed many kinds of artifacts that happened during library preparation that had always been invisible, had never been seen before, because the fidelity was never sufficient to even see these low-level artifacts. So we also spent about 2.5 years optimizing our library preparation, how we process the DNA in the lab, in order to remove those artifacts, until we got to a point that there was no discernible artifact pattern. And essentially, these random errors can then be filtered computationally, and I'll show you in a second what that pipeline looks like computationally. And we can then detect double-strand mutations, which are those that are present those changes that are present in both of the strands from single strand mismatches and damage that are present in only one of the two strands. So this allowed us to achieve for the first time single molecule sequencing that also has single molecule fidelity. So we can believe things that we only observe in a single molecule. We don't need a multiplicity of molecules in order to convince us that these changes are real. And I'll show you the data that convinced us indeed we reached single molecule fidelity. For the bioinformaticians in the crowd, I won't go through every step here, but just big picture, we did have to develop a new computational pipeline to analyze this data. That was this, I would say, just as important as the experimental side of this project, because we had to implement a series of filters that remove computational artifacts. It turns out that even with the absolute best, cleanest sequencing we could have hoped for, computational artifacts also rear their head and show up when you're dealing with such rare phenomena. So, for example, there were a set of filters that removed, quote unquote, low quality molecules or bad molecules, those that, for example, we didn't have a good quality consensus for both of the strands, or there was a missing adapter in one of the ends. We actually found out that aligning to the T2T, telomere to telomere genome, reduced artifacts as well, because we removed some misalignments. We then had to drill down to filters at the base level, removing regions of the genome that are prone to artifacts, regions with segmental duplications, for example, certain kind of repeats. These kinds of filters would be familiar to anyone who does somatic mosaic research, I would say. But we did have to fine tune some special filters for this data. For example, base quality. There's a certain measure of consensus-based quality in PacBio sequencing that turned out to be very, very important. Then we filter out those variants that are seen in standard sequencing of that same individual to filter out germline variants, those that were inherited by the parents, so that we're then left with only somatic mosaic mutations. And, approximately half of the data is filtered out by the time we get through this whole process. So this is what raw data looks like for just one molecule. And you're seeing in purple at the top, each row is an individual pass of the polymerase for the top strand, and in pink is the individual passes of the bottom strand. So you see how many times the top strand was essentially copied by the polymerase independently, and likewise for the bottom strand. And you can see all these random sort of colors sprinkled throughout. These are mismatches from the reference genome. These are essentially a lot of this random sequencing errors that occurred. And you can see how computationally it's like really easy to filter out these random sequencing errors. And in some locations, you see a streak of color going down vertically all the way down. It's detected in almost every pass of both the bottom and top strands. These are high confidence calls of differences from the reference genome. And at the bottom panel is standard DNA sequencing from that same sample. So you can see everywhere that there's a vertical streak in that single molecule, you can also see that variant was also detected in the standard germline sequencing of the individual, except for one location here. And that's what we would call as a mosaic mutation. It was detected in this single molecule with high confidence, but it wasn't detected in the standard DNA sequencing of the individual. And this is what it looks like zoomed in. So how do we really know what our fidelity is, that we reach single molecule fidelity? So essentially what we set out to do, and now and others have done this too, which is take a sample where we know through independent methods what the expected mutation burden is, and more specifically, or ideally, take a sample that has the lowest possible mutation burden to really put our technology to the most rigorous test. And sperm have both of these properties. Sperm, through de novo mutation studies, where you count the number of mutations in an offspring that were not detected in the parents and can be phased to the father, gives you an estimate of the number of mosaic mutations that were in the sperm that formed the child. And in addition, sperm actually turn out to have the lowest mutation burden of any accessible tissue other than eggs. So we basically took a set of purified sperm samples, and measured them with our method that I forgot to mention, we called high-def-seq. We simultaneously measured those same samples with a form of duplex sequencing called nanoseq, and we compared them to the expected paternal de novo mutation rate from other studies. And these all showed concordant results, and you can even see the detection of the slight increase in mutation burden in sperm from older men compared to younger men. And because we know from other studies that mutations increase with age linearly, we profiled a set of tissues of individuals from different ages and successfully detected this linear increase with age. And so far, I should just be very clear, we're only analyzing double-strand mutations in the data I'm showing in this slide. So first, we're setting out to see, can we detect double-stranded mutations, meaning mutations that are changes that are present in both strands, before we then look at single-strand changes with this technology. So seeing this linear increase, tells us that our noise level must be lower than the noise level at the youngest ages here. Otherwise, we wouldn't have seen this linear increase with age. So once we established our fidelity for changes in DNA that are in both strands, i.e. mutations, we then looked at the data and asked, can we find high confidence changes in single molecules that are in only one of the two strands? And this is what one of those examples looks like. You can clearly see the streak of color in only one of the two strands, not in every pass of the molecule, but this is very high confidence detection. And now the question is, are these real? Are they artifacts? Are they artifacts of the sequencing, artifacts of the library preparation? How would we figure that out? How would we convince ourselves these are reliable? So we reasoned that individuals whose healthy tissues are known to have higher than normal double-strand mutations would also have higher than normal single-strand call burdens. So we collected a set of samples from individuals with cancer predisposition syndromes. These are individuals that have higher than normal mutation rates. That's actually why they're predisposed to cancer. And then we measured the single-strand call burden. And we found that in two categories of cancer predisposition syndromes, we indeed can detect an outlier high single-strand DNA call burden, specifically in individuals with biallelic mismatch repair deficiency and individuals with defects in polymerase proofreading. So the polymerases that copy their genome during replication don't proofread well. So they have abnormally high mutation burdens. And so we see a correspondingly high single-strand call burden. But just the burden, obviously, it wasn't enough. We then looked at the sequence contexts, the signatures, the sort of patterns of both the double-strand mutations we saw in these samples and the single-strand calls, and then we compared them to each other. So this is one sample, an example, from an individual with polymerase proofreading defect. At the bottom, you can see which sequence contexts tended to have mutations. For example, TCT to TAT was a very common mosaic mutation seen in this polymerase proofreading deficient individual. And actually that pattern was known from cancers from these individuals. And looking at the single-strand calls, we actually see a peak of single-strand calls in that same context, which really definitively shows us that these single-strand calls we're seeing are precursors of these double-strand mutations. And not just that, but we don't just see them at the TCT context. We can actually distinguish between the two strands because we're seeing single-strand level data. We can actually see that it's AGA to ATA changes that are prevalent, not TCT to TAT. And then the AGA, ATA on one strand then are fixed into what we call TCT to TAT, but essentially once there are mutations, you can't distinguish whether it's the TCT to TAT, or in other words, AGA to ATA, came from a TCT to TAT change or from an AGA to ATA change. So essentially, once you observe double-strand mutations, you have no way to know which of the two strands tends to be the one that has a mismatch or a damage to give rise to that observed mutational pattern. But when we reveal these single-strand changes with high-def-seq, we can now distinguish those two. And there was evidence from prior studies, indirect evidence from replication timing experiments, that concluded that this is the strand that's likely giving rise to this mutational pattern. Here we're observing it directly. We profiled tumors from individuals with these cancer predisposition syndromes, specifically hypermutating brain tumors. Individuals with biallelic mismatch repair deficiency tended to get brain tumors that the brain tumors then have a secondary somatic mutation in polymerase proofreading. So these tumors are defective in two different ways. And so they have extremely high mosaic mutations, which you can see here with levels upwards of 1 in 10,000 base pairs. 1 in 100,000 base pairs, which is actually really high. And they had a correspondingly higher single-strand DNA call burden. And the tumor that had a higher mutation burden also had a higher single-strand DNA call burden. And again, looking at the patterns, we see this remarkable correspondence between the double-strand mutation patterns and the single-strand call patterns that also then tell us which of the two strands is the one that actually the mutations originate from. So we compared our single-strand call burdens also between the sort of up until that point, the gold standard method of duplex sequencing called NanoSeq, and specifically for sperm. And you can see that at the single-strand call level, there really is essentially almost no single-strand calls in these healthy sperm samples other than C to T changes. Whereas in NanoSeq, there are a bunch of C to T changes at a higher level, but also a bunch of other single-strand changes. So that tells us that pretty much almost all single-strand changes that are seen in standard duplex sequencing that involves amplification of the DNA before sequencing through PCR has a bunch of artifacts, probably from the PCR itself, that is avoided by directly sequencing single molecules without any amplification. And this is sort of a comparison of double-strand mutation burdens for a bunch of samples that we profiled both by our high-def-seq method and by NanoSeq. Very strong correlation, but then at the single-strand call level, high-def-seq consistently has much lower single-strand DNA call burdens compared to NanoSeq. So now we, more recently, we've become very interested in looking at these single-strand call burdens in healthy tissues, so not just in cancer predisposition syndromes. And so far, we actually found one tissue type that's very surprisingly has an outlier single-strand DNA call burden, and that's sperm. They have an outlier fraction and burden of single-stranded C to T changes. And that's surprising because at the mutation level, sperm have the lowest mutation burden. So how is it that a sample has a specific sample type, sperm, have the highest single-strand DNA call burden out of all healthy tissues we've profiled, while also having the lowest mutation burden? It's sort of a discrepancy. And that's highlighted here. And so we looked at the pattern of these C to T single-strand DNA changes in sperm, and it turns out that they matched very closely to a completely separate experiment that we did, where we heated up DNA in the lab from healthy blood, because we thought that heating up DNA would deaminate cytosine and show us the pattern of heat damage to DNA that happens in the lab. So heating up DNA at 72 degrees C for 6 hours gave almost the exact same pattern as what we saw in sperm that was not heated. It was actually processed very, very gently. And so, of course, we thought, well, maybe something about how we're extracting DNA from sperm is causing this. And so we went through a very long set of control experiments and concluded that this is present in the sperm DNA and is not due to our processing of it in the lab. For example, we took cells and blood DNA and pass them through the exact same process we pass sperm purification through and sperm DNA extraction through, and we did not see this pattern. Essentially what we think is happening here is that sperm have a higher burden of cytosine damage, cytosine deamination into uracil that then is sequenced as C to T changes, probably because sperm sperm DNA in the final stages of sperm development is very highly compacted by protamines, such that the DNA is not accessible to DNA repair. And there's really not much active DNA repair going on in the final stages of spermatogenesis. And so that presumably, at least we hypothesize, allows DNA damage to accumulate in sperm, which then after fertilization is corrected by the DNA repair machinery of eggs. So that damage never becomes mutations in the next generation in offspring. That's our working model. Just to show you a little more of what we can do with this technology, we not only can sequence A, C's, T's, and G's, but we can also recover information about how long it takes to incorporate each individual base as the sequencer polymerase goes around and around the molecule. It's called kinetic data. And we actually have two types of kinetic data. One is called the pulse width, which is the amount of time that the polymerase takes to incorporate a base, and the interpulse duration, or IPD, which is the time between base incorporation events. So we actually looked at these sperm single-stranded DNA C to T changes, and we thought, well, if these are indeed not really thymines, they're actually uracils, they should have a different kinetic pattern than thymines at a location of what normally is a cytosine. And indeed, that's what we found, that in the +1 and +2 base position relative to the C to T changes in sperm, there's a kinetic difference compared to true C to T changes, which we know are C to T because they're double-strand mutations. And when we scramble molecule labels and as a sort of control, those patterns go away. We also treated sperm DNA and blood heated up in the lab with UDG enzyme, which excises out uracils, and those C to T changes we saw in sperm DNA went away, confirming that they are a type of uracil. There are different types of uracil damage. At least there's some type of uracil damage. We also profiled mitochondrial DNA with high-def-seq, and we were interested in whether we might figure out something new about how mitochondria mutate. What's interesting, and this is very well established, and we confirmed this with high-def-seq, is that at the double-strand mutation level, the mitochondrial genome has a significantly higher mutation burden than the nuclear genome, between 40 to 60-fold higher, which you can see here when we measure this in liver and kidney. So we thought, we almost expected, well, maybe if most mitochondrial mutations are arising from sort of lack of fidelity of mitochondrial genome replication due to mismatches that are incorporated, we might see a much higher single-stranded DNA call burden in the mitochondrial genome relative to the nuclear genome. But in fact, we did not see that. That suggests that the mitochondrial genome is mutating by a type of damage that we do not detect, not by canonical mismatches, but some kind of damage we don't detect with our method. And even more so, it suggests that that damage is happening during mitochondrial genome replication. So it's very quickly being fixed into double-strand mutations. So we never see it with high-def-seq, because in high-def-seq, we don't profile single-stranded DNA during replication that is not yet replicated. We only can profile double-stranded DNA. So we'll never see transient events that are single-stranded damage then quickly become double-strand mutations. ApoBec3A is another sort of endogenous type of DNA damage. Is really fascinating that we wanted to study with high-def-seq. So our body has these enzymes that evolved to basically mutate incoming viral nucleic acids, but they have off-target effects on our own genome. And in fact, they've been shown to sometimes be cause an abnormally high number of mutations in the setting of cancer. ApoBec3A was relatively recently identified to be one of the pretty much the key ApoBec enzyme that mutagenizes the nuclear genome. And so we took fibroblasts and expressed ApoBec3A in them, and we measured whether we could detect directly the single-stranded DNA damage caused by ApoBec3A in addition to the mutations caused by ApoBec3A. And indeed, that's what we saw. You can see here the single-strand DNA call burden was higher in the setting of ApoBec3A expression. And we looked at the pattern of C to T changes. I should mention that ApoBec3A, the type of damage it induces is cytosine deamination, similar to the type of damage we saw in sperm, but it's with a different sort of pattern of sequences than the pattern we saw in sperm. And so you can see here the pattern of single-stranded DNA C to T changes we saw with ApoBec3A expression, and that matched very closely to the known ApoBec3A double-strand DNA mutation pattern, but with an exception. You can see these additional sequence contacts that we saw very clearly and reproducibly in our data that have not been seen at the level of mutations in analysis of cancer genomes, for example. So it's not clear yet, what these what explains this discrepancy, but one possibility is that the damage caused by ApoBec3A is repaired more efficiently in certain sequence contexts than others. So for example, if these sequence contexts highlighted in this black box here are repaired more efficiently in vivo, then those would not become double-strand mutations relative to these 4 other sequence contexts that are not repaired as efficiently, and then they would actually show up as mutations. And so we've started to sort of create a catalog of single-stranded DNA damage and mismatches that we're giving names that are similar and corresponding to the names that are given to mutation patterns. And sort of our long-term goal is to relate these single-strand DNA patterns to double-strand DNA patterns. And so I'll summarize and then also talk more about this sort of bigger idea of how to relate single-strand and double-stranded patterns in order to study processes of mutation. But just to summarize first, all human tissues accumulate mutations with age. Measuring these requires ultra-high fidelity sequencing methods. And we've developed a method that can resolve double and single-strand events in single molecules. And hopefully I've shown you sort of the beginnings of how we can see single-strand changes and how these can teach us something about origins of mutations. Future directions, we would like to apply this technology to study other aging phenotypes and diseases. We'd like to maybe detect the single-strand precursors of insertions and deletions, or maybe even structural variants. I've shown you only analyses so far of single base substitution at the level of single strands, but of course the same kind of idea would apply to insertions and deletions and maybe structural variants. Because we have relatively long reads of 1 to 4 kilobases, it could allow us to study processes of mosaic mutations in regions of the genome that are not accessible to short reads. So that's another sort of application that we're interested in. And I mentioned, and now I'll talk a bit more about our long-term goal of building a comprehensive catalog of single-stranded damage and repair patterns. So I showed you a few examples of single-strand patterns that we've seen. For example, on the left side, the top is the pattern that we saw in polymerase proofreading deficient samples. In the middle is the pattern that we saw with ApoBec3A. And the bottom is the single-stranded DNA damage pattern we saw in sperm and when we heat up DNA in the lab. So we're seeing all these patterns. And there's also known patterns on the right of mutations that are seen in healthy tissues, in cancer. And so the question is, how are these related to each other? Not just in terms of like their direct similarity, but also conceptually. So it's important to recognize that there can be discrepancies between these patterns because what comes in the middle between the process of mismatches and damage is DNA repair and replication. that itself can have biases. For example, as I mentioned, it could be that certain contexts are repaired more efficiently, and that could give rise to a discrepancy between a single-stranded pattern and its corresponding resulting double-stranded mutation pattern. So if we eventually collect a whole bunch of single-stranded signatures, whole bunch of double-stranded mutation signatures, we may be able to deconvolute and deduce DNA repair and replication signatures. Of course, there are also experimental methods that we could apply to try and figure this out. Another sort of conceptual way of thinking about this sort of new type of data, of single-stranded mismatches and damage data relative to mutation data, is along the temporal axis. So imagine this is all schematic. On the left are burdens of single-stranded DNA mismatch and damage over time. So for example, you might have a constant single-stranded DNA damage process in red over time, and you may have episodic damage events of other types. Maybe someone was exposed to a hefty dose of radiation, or someone ingested some kind of carcinogen at that time point in yellow, for example. And then in the middle, you can see sort of what would the single-stranded DNA mismatch and damage pattern be after DNA repair and replication eliminated some fraction of that mismatch and damage. For example, maybe a certain fraction of the red process is consistently repaired successfully. And so its level is brought down. Likewise, for the green process and the yellow process, and maybe the yellow process is more efficiently repaired than the green process. But then what do we actually see when we profile mutations? If theoretically you could profile mutations at every time point, you'd see something like this. You'd see a gradual accumulation of mutations resulting from the red process. You'd see a sudden jump in the green mutation process, and then a sudden jump in yellow, in the yellow mutation, the mutations resulting from the yellow process. And so you can almost see this mathematically, that mutations are really the integral of the leftover unrepaired or replicated before repair single-stranded DNA mismatches and damage. And so if imagine you don't have data at every given time point, imagine you profile the sample right at the beginning of where this yellow process was accumulating. I think you can see my cursor, hopefully. Imagine you could have a sample right here. You wouldn't actually see yet the accumulation. You wouldn't even see that there was a yellow mutation process, because it hasn't had enough time to accumulate yet. But if you were to process that sample at the single strand level and reveal those single strand changes right at that moment, you may actually see clearly that single strand process is happening even before it had time to accumulate over time. So single strand DNA profiling could reveal mutagenic processes in the moment they're happening, and without having to wait a while for those mutations to accumulate to a sufficient level to then be able to deconvolute their pattern from the mass of other mutations. Conversely, if you profile a sample after these episodic processes are happening, for example, right over here, you wouldn't see that at the single strand level any evidence of this yellow process, but you would see it at the mutation level, because the mutations are a record of prior mutational processes. So putting it another way, mutations reflect the sum of prior imbalances between new lesions and repair. Single-stranded DNA profiling reflects the current balance between lesions and repair. And this is what could allow us to use single-stranded DNA profiling to interrogate dynamics of the system. Because it's looking at the real-time status, DNA damage and repair may not always be in equilibrium, and if you can push the system out of equilibrium through any kinds of experimental methods, we could then resolve dynamics of the process, which you couldn't do by profiling only double-strand mutations. So with that, I just wanted to acknowledge the really, I would say, brave members of my lab that took on this project because of its many technical challenges along the way. Mei-Hong Liu and Ben Costa, and there were others in the lab as well that contributed. We had a really wonderful group of collaborators: Jonathan Shoag at Case Western, Uri Tabori at SickKids Hospital, the Cryosperm and Egg Bank, Richard Wagner from the Universität Sherbrooke, and the NIH Neurobiobank. And we also had the support of our sponsors. Happy to take questions. Thank you.
Thank you. Thank you, Gilad. So yeah, I saw Ludmil have a question, so he could go first.
Sure.
So sorry, I just put my hand up. Thank you for the beautiful talk. I mean, high-def-seq is really quite interesting and quite remarkable. What I find surprising, and I found surprising in your publication, is you focus very much on endogenous processes. And I was wondering, because I would have expected to see UV light, benzoapirin, things that where we really know the damage on one strand.
Yeah.
Have you done that? Have you observed that as
That is something we're very, very interested in. It's a great question. There are actually two technical considerations, however, for that. One is types of damage where the sequencer polymerase that's used by PacBio sequencing that cannot get across, we won't see those molecules. That's one. And we know that to be the case for some DNA damage already. That's one consideration. The other is that currently, in order for us to see that damage, it has to lead to sort of a significant enough change in either the base pairing or in the kinetics. And so that's something we're interested in is what are those types of damage that we could detect. So for example, cytosine deamination, it was very, very clear. It leads to a mismatch. It makes its cytosines look like, and it makes uracils look like thymines. So very easy to detect. Other types of damage may not be as easy to detect. And so that's an open question: to what degree do different types of damage show up in the data and can be reliably called? But then there's the secondary issue of, can the polymerase even get across that damage, or just get stuck and falls off? And if the polymerase falls off, that we're never going to see that molecule in our data.
Got it. And have you tried to see if it falls off with UV light or BAP?
Yeah. So I mean, so far it does seem like thymine dimers are not able to be sequenced reliably. I should mention that there's also other groups, trying to detect damage with Oxford nanopore sequencing, and that's a different kind of obviously different technology. There's no polymerase, so it's just a pore. So it's not as likely to get stuck. On the other hand, with Oxford nanopore sequencing, you don't get this repeated measurement over and over and over of the same molecule. So the fidelity of that is less than what we're doing. So there's a trade-off there.
Yeah, I think ONT is potentially great for some damages. I wasn't sure whether this can be used. I have other questions, but I'll leave other people to ask them. I just want to make one comment. What you show in the apobex, that pattern of CCCs in the middle, we do see it in certain cancers, and there has been a hypothesis is that it's one of the human glycosylases, like NEO1 or 2, that may be causing it. So it might be interesting for you to check that. But it's seen in a small number of cancers that they will actually amplify that middle peak.
Interesting. Yeah, I'd be very interested. I have heard from two people so far that said they've seen hints of this in some situations. And Mia Petliak, who's actually here at NYU, has also mentioned. So, yeah, I mean, I think it's really interesting. It's interesting probably to figure out why in certain situations this might show up as mutations and other times it may not. But all I'm showing, like, at least in this system, very simple system of overexpression of apoBacteria and fibroblasts, there was it showed up very clearly.
Yeah. Thanks again for the talk.
Thank you. And thank you, Ludmil, I should say, for launching the field of mutational signature analysis. It's changed genomics forever. So yeah, I see Mike. Yeah.
I think there's a question in the chat actually before I raise my hand.
Oh, sure. I see one in the chat. So it says, I'm wondering about the limitation of high-def-seq. This is from Changuk. What was the minimal DNA amounts you tried? Minimal genomic regions covered? And does it include STR regions as well? Indels are not described. Okay, sure. So amount of DNA we need, we were able to get it down to about like even 400 nanograms, but it depends on the quality of the DNA. That's high quality, high molecular weight DNA. We found that if DNA is fragmented, for example, post-mortem DNA, you actually need more of it. And then we do a modified library preparation that removes artifacts that occur from post-mortem fragmented DNA. And in those situations, you might need as much as 1,000 nanograms to 1,500 nanograms. So it's not something you can do on single cells. It's only a bulk assay. The other limitation is, what fraction of the genome we're covering. So in the first version of high-def-seq, we use restriction enzymes to cut the DNA. And then we basically capture about 30 to 40% of the genome, which is enough to see pretty much most patterns, but doesn't allow us to see any genomic regions that are not in that 30 to 40%. However, since then, and actually we snuck it into the paper right before it was published, we did develop a variation of high-def-seq that can profile any genomic region with random fragmentation, not with restriction enzymes. It just requires about double the amount of input DNA. As far as STR regions, we do have to filter them out because they're prone to indels. And those then can give rise to false positive single base substitution calls. We don't filter out all of them. We're actually quite conservative about how much we filter out, but we do need to filter out some of them. And indels, we've not yet been able to reach single molecule fidelity because you saw how we can really filter out errors because they're random when it comes to single base substitutions. Indel errors of the sequencer are not actually random. And because of that, even though we read the molecule over and over and over, you can get recurrent errors in the same position, and that leads to false positive calls. So we were very interested in indels. We're still working on it, but it's been much more challenging than single base substitutions. Yeah, Mike.
Thanks for the great talk. So I was curious, since you're using PacBio here, you could also read out methylcytosines. And I was curious if you thought about ways to incorporate that information into your analysis. For example, you could include as like a fourth base in your, or sorry, a fifth base in your signature analysis, and try to understand like patterns, how methylcytosine affects, patterns of mutations in the context of methylcytosine.
Absolutely. So yes, we could do that. We haven't done that, but it is definitely something we're interested in doing. The detection of 5-methylcytosine actually occurs through the same kinetic analysis that I showed you. So we're basically trying to fine-tune the kinetic analysis in order to recover that information. And then yes, we can layer on simultaneously methylation and all of this other data simultaneously. So yeah, absolutely. And there aren't, as I mentioned before, known influences of methylation on mutation. And it might be regional also. It might not just be at the specific base that's methylated. There's some data in the literature suggesting that as well. Because, for example, methylation might change the accessibility of the DNA to DNA repair. It might change what proteins bind to that region of the genome, which then may affect damage and mutation.
Right. Thanks.
So yeah, so Joe is asking, are there some types of mutations that are not random in PacBio sequencing? Are there any SNVs that are non-random? So there are up to, but those become an issue when you don't have enough passes around the molecule for SNVs. Because we have so many passes, generally like 20 plus passes, that's sufficient to remove even the low strength of non-randomness SNVs. Indels, however, as I mentioned, are definitely non-random in PacBio sequencing, and those, even with 20 plus passes per strand, are still challenging to remove. But we do not see any discernible artifact pattern of a non-random SNV sequencing errors that survives after the requirement of 20 passes per strand. So yeah, hopefully that answers your question. In fact, 5 passes per strand is enough to get enough confidence for double strand mutations. for single strand changes, you really do need sort of 20 plus passes per strand. Great, I don't see any other questions.
Thank you, Gilad. I think it's about time. So if anyone have any other questions, feel free to email him. He has his email in the poster. Thank you.
Thank you very much for hosting me. Thanks.